So what we're going to look at in lab on Wednesday is ultimately protein folding and unfolding. Okay? So your lecture professor will likely not get into protein chemistry, which means we aren't going to get into it too much. This lab is mainly designed to just kind of give you an overview of what you might expect to see in biochemistry if you have to end up taking that. Okay? So first and foremost, a lot of biochemists that I've ever talked to, including myself, would have made the claim that organic chemistry is unnecessary. You don't need to know it for biochemistry and not particularly relevant. And to them, I really say they should look at a dictionary. Because organic chemistry is the study of molecules made up of what? Carbon and hydrogen, which is the definition of organic. Okay? Organic also happens to refer to living things. So if we're going to look at biochemistry, which is the study of living things, you are looking by definition at organic chemistry. Okay? And so what ends up happening is that biochemists get bogged down in all the names and terrible nomenclature systems that they end up generating, that they tend to ignore the fact that it is ultimately chemistry, and that a lot of the intermolecular forces and a lot of the things that drive the reactions that they're studying in biochemistry are rooted and based in organic chemistry. Okay? The primary thing that's going to drive a lot of this is looking at intermolecular forces. So you're looking at ionic bonds, hydrogen bonding, your dipole-dipole forces, and then London dispersion forces. And those will drive and determine a lot of interactions between individual proteins and DNA and biological systems. Okay? So if you know your forces, you can apply those forces in a much broader sense to biochemistry and do better as far as the reactions and understanding things when you move on. Okay? So we just kind of talked about this. It is relevant to biochemistry. Okay? It's almost all found through organic chemistry. Okay? So we're really looking at three main branches when we move into biochemistry. You've got DNA, RNA, and proteins. Okay? And depending on who you talk to, they may decide that one of those is more important than the other. I don't particularly care about the fight. They can do whatever they want. It's still organic chemistry. Okay? So as far as complexity goes, they all have their own little quirks and nuances. And what we're going to try and do is look at proteins and very simple proteins and see how their environments can end up changing the physical properties of those proteins and what things we can visualize from it. Okay? So as far as our fields, we're going to jump into proteins. So the protein that I ended up showing was probably one of the more important ones. We're looking at ATP synthase. It generates energy for all life. Okay? We end up generating ATP, our storehouse for all energy. Comes out of that molecule. Okay? That molecule or that protein is composed of several other proteins to form that overall structure. And it's those individual units and how they arrange with each other or organize that ends up generating the final structure that has so many important properties. Okay? So what we could go through and do is actually look at an individual case. So those little green helices shown, okay, what we've got in here is actually a combination of 12 different individual proteins that have all independently folded and then interacted with each other to give that overall structure. Okay? If we wanted to take it further, we could go ahead and look at the protein data bank. Let's see, can I scroll nicely on this? Oh, look at that. And I can't zoom worth junk on this, but if we go ahead and take a look at this individual link, it shows a variety of different images. You see some arrows, some squiggles, and all that fun stuff. Those are representing a secondary structure. Okay? If we look even into the smaller print, you can see a sequence of letters. Okay? Those letters represent the individual amino acids that made up the overall protein. Okay? So what they tried to show with this is just the information to make one sequence of proteins. Right? We then scale that up 12 times to get the bulk unit, and then we have to scale even further to get the rest of that protein structure. And you can tell that it looks fairly complicated and difficult to interpret. So what we want to do is start to learn something about those individual units and how we represent them and simplify them. Okay? So if we look at just the sequence of letters, what we're referring to is the primary structure of our protein. Okay? So it's just the sequence of amino acids. And if we tried to draw out the individual structures, we'd probably find that it's going to take up pages and pages of text and just be too difficult to evaluate. So what they end up doing is instead of looking at the entire structure, they only look at the pieces that they feel are important, which would be our side chains. Okay? And they assign those letter codes. Each of those amino acids is linked to another amino acid through an amide bond. That amide bond is highlighted in red. Okay? And what we're looking at is the linkage between a carbonyl and an amine. When we connect those two pieces to each other, we end up forming the amide bond, and that's going to generate our backbone. Okay? This sequence should look really, really familiar because we've already seen this before. Where have you guys seen this before? Say maybe last week. It's a polymer. Proteins are polymers, as magnificent and amazing as that can be. Okay? What we're looking at is a complex polymer sequence that biology has put together to generate certain properties. Okay? So what we're looking at is individual compounds or monomers being put together to generate our protein backbone. Okay? Our physical properties that are going to determine how our amino acid interacts or our protein interacts with the rest of the world comes in with these R groups, which you'll notice are interspersed on our protein backbone. Okay? Those are what's going to interact with our outside world and generate the shapes and structures that we end up seeing. Okay? For each individual R group, we could have a huge variety of different pieces. Okay? As far as life is concerned and biology is concerned, there ends up being 20 common amino acids. These are the primary amino acids used to generate pretty much everything that we see and breathe in the organic world. Okay? Those R groups are highlighted in blue, and you'll notice what's highlighted in pink is kind of separated out to the sides. What's the difference between the things highlighted in pink? Nothing. Everything in pink is the same all the way through that. Okay? They're all the exact same thing. That's because that's what's generating our backbone, okay, our polymer backbone. So we aren't too concerned about the reactivity of that because that's buried inside the backbone. What we're concerned about is what's hanging outside of the backbone. And that's where we're looking at our R groups. Okay? So if we take a look at our first one, that upper left, we've got a CH3. If we compared that to, say, the one right below it, okay, would we expect different properties between those two R groups? Okay, what types of bonds are in each of those? Are they ionic bonds? Are they polar covalent? There we go. At least I'm getting some kind of answer. We're looking at covalent bonds. If they're covalent bonds, what does that mean about the types of intermolecular forces? They're the same type of intermolecular forces because it's the same type of bond. What type of intermolecular force would we be looking at? London dispersion forces. Okay? So if we looked at those two compounds, we would predict them to have very similar chemistries. Okay? Their shapes are different, so we would expect slightly different interactions, but for the most part, very much the same. Okay? But what if we compared those to, say, something down at the very bottom? Say that R group. Okay, aspartic acid. Would we expect a different interaction now? Good answer. Yes, we would expect a different interaction. Why would we expect a different interaction with aspartic acid? Because instead of being just covalent bonds, we now have polar covalent bonds, which means we generate a different intermolecular force. That different intermolecular force is what's going to cause a different interaction with the rest of the environment, and we're going to see different properties come out of it. Okay? And of course, since we have different things, what are we going to have to do? We're going to name them differently. So I already started to use some names. For that bottom one, we're looking at aspartic acid. Why might we jump to a name as opposed to using the structure to show the differences? Because now we're biochemists and we can't understand structures. We need to look at names. Okay? It is, but as far as I'm concerned, that simple. Okay? So they use aspartic acid as a name. Okay? But not only do we not like names as biochemists, okay? Just like with organic chemists, we didn't like dealing with names. We have to shrink it down to something even smaller. Okay? So we come up with 3-letter codes. So for aspartic acid, we get ASP. Okay? Because even that's not short enough, they shorten it down all the way to a 1-letter code. Okay? In this case, D. What does that mean as far as what you need to be able to do as a biochemist? You need to know the structure, the full name, the 3-letter code, and the 1-letter code for an individual amino acid. That's a whole lot of useless information, okay, that you have to have memorized to be able to process when you move into biochemistry. Okay? What do I expect out of you as far as organic chemistry goes? Of course, more useful information. So I would expect some structural information. I want you to know what's going on with that structure, and in particular, how can it interact with something else? Okay? That interaction is ultimately what we're looking at when we look at our classifications. Okay? So there's several different ways that we can classify our amino acids. You need to be able to look at those individual structures and process what does each of those amino acids have as far as its polarity, its non-polarity, acidic, basic, all of that fun stuff. Okay? So if we took a look at our top two, alanine and valine, or valine, how might you classify those?
Non-polar hydrophobic. Sorry.
We might expect to classify those as nonpolar and hydrophobic. Why would they be nonpolar? Because there's no polar bonds. Why are there no polar bonds? There's no electronegative elements. Okay? Why would we expect them to be hydrophobic? Well, hydrophobic means what? Scared of water. What do we know about water? It's polar. Well, if our compound's nonpolar, probably very likely to not interact with water. Okay, so we'd be looking at something that's hydrophobic. Okay, so being able to evaluate those structures is really important. Okay, because some people do have to move into biochemistry and will be required to know this information or know all of those amino acids, we're going to start to push you down that path. I expect you to have 3 memorized. Which 3 do I expect you to have memorized? I don't care. Pick 3 and memorize those 3. Okay? Start that process of trying to get that information contained so that you can then process and use that when you move on into other chemistries. Okay? Or for that matter, dietetics. Okay? Pretty much any field that you could deal with is going to deal with proteins. Okay? Once we start to combine those structures together, what they ended up noticing is that we don't get just a long chain of amino acids. Okay? We don't just get a polymer protein. Our structure changes and tweaks in very particular patterns. Okay? So what's shown in the middle in both of these cases is roughly what you would expect to see given the complete structure. Okay? Except even in these cases, we've got it simplified. Instead of actually showing what those R groups are, we're just saying there are R groups. Okay? And if you stare at that structure long enough, okay, or try to simplify it, you might start to notice that we get this kind of spiral-looking shape coming out of it. Okay? And what we've generated in this case is an alpha helix. Okay, so this is one of the secondary structures that can come from proteins. The other option is we could get beta sheets. So our protein chain aligns in such a fashion to neutralize the intermolecular forces between our compounds. Okay, and we end up getting sheets, okay, or rows of proteins chains aligning with each other as opposed to coiling. Okay? Those interactions are primarily due to the side chains interacting with each other. Okay? So if we have a very particular sequence of amino acids within a protein, we can end up seeing an alpha helix, or we could see the beta sheet. Without that proper sequence, we don't get those patterns showing up. Okay? So our side chains are really, really important to determine that secondary structure. Last thing that can happen is when we move into tertiary structure, and this is where our solvent plays a much larger role. So depending on what solution we're in, we're going to change how our protein then, our secondary structure then folds upon itself. Okay? So what we're looking at is pretty much a new interaction. Okay? That new interaction is going to be our nonpolar, or based in our nonpolar side chains. Okay? We're looking at a biological system. Biological systems are primarily found in what solvent? Water. Okay, what can you tell me about water? It's polar, which means if we look at our amino acid structure, all of our nonpolar groups that we already said were hydrophobic are going to try and run away from water, okay, to avoid that interaction, much like when we added diethyl ether to water. We don't see them mixing. We see two separate distinct layers. Okay, but since our protein is still connected to some polar groups, what ends up happening is a hydrophobic interaction that causes our nonpolar parts of our protein to fold inwards, okay, so that they can be in a nonpolar section. What ends up being on the outside of our protein? All our hydrophilic interactions, or all our hydrophilic amino acids. Okay? So it's that complex folding that ends up generating our tertiary structure, okay, which is primarily coming from our solvent. Okay? Depending on the fields you move into, or you could look at, if you moved into biochemistry, you could look at the chemical aspect or even the physical model. In this case, we've got, kind of skip through it. This group took a very simple protein structure. So in this case, it's just that single strand. And they then decided to model the different interactions that came out of that. So they let the structure flip and rotate on its own, and you'll notice everything's moving rather violently. Okay? And you can see an alpha helix start to form that stays fairly constant. And if you look really carefully, every so often, you'll see a beta sheet start to show up as well. And so all they're doing is modeling those individual interactions, the intermolecular forces, trying to stabilize each other in some fashion to generate our 3D structure. Okay? Because we don't need to watch this whole thing, we can skip to what I think is the punchline. Keep going. Where is it? So that was their average. They took those and averaged their structure. And what they've got right here is they took the actual three-dimensional structure that someone obtained using X-ray crystallography. So ultimately an actual three-dimensional image. And they compared that back to their modeled system, okay, using computers to correlate how the intermolecular forces cause the structure to overlap. And what you'll notice is there is some cases where you see two images, okay? And that's because their image wasn't absolutely perfect. But if you consider, one was the absolute naturally made protein, and one was a sequence of data put into a computer to generate a randomly oriented structure. And they ended up coming up with something virtually identical. Okay? So it's the power of prediction from the intermolecular forces and how they interplay with each other that's ultimately what's driving this. Okay? I personally think that's kind of cool to see that someone could actually put this together. Okay? And use just their predictive skills to be able to figure out what happened. Okay? So that's an example of our tertiary structure. As we move even further up, we can move into quaternary structure. And in that case, what we end up doing is taking several protein units, and those protein units will stack and fold with other protein units to generate the quaternary structure. Okay, so in this case, we've got our ATP synthase. Okay, so we can look at a video of ATP synthase moving around. It makes it look like kind of a giant brain. But what's happening up at the top is ATP is moving in and out of this. you look at the very bottom, there's something rotating there. Right? We'll look at that rotation or what's causing that rotation in a second. They're now going to zoom in so you can actually see the amino acid side chains that are generating the structure. And what's about to float in is a structure of ADP and a single atom or a single molecule of phosphate. The protein grabs and holds those two pieces and forces those two molecules near each other. Right? The instant they're close enough, what ends up happening is those intermolecular forces form bonds, and we end up physically forcing our reaction to occur. Okay? If we zoom in at the very bottom of this chain, what we've got here is a proton gradient. So those little tennis ball looking things are hydrogen atoms that are getting pushed through a membrane, and in the process of getting pushed through the membrane, it causes that blue unit to rotate. As it rotates, it causes the orange unit above it to rotate, which then forces individual protein units above it to then bend and flex, opening and exposing different sections of protein to allow for the formation and reaction of ATP. Okay? So it's an amazingly complex system that generates just a simple single molecule that drives pretty much all life and energy as we know it. Okay? As fun as it would be to actually analyze that in the lab, we don't have those capabilities. So instead, what we're going to look at is isolating a protein from spirulina. Okay? This particular protein happens to collect solar energy. So what its job is to absorb energy and then transfer that energy on through the electron transfer pathway to generate ATP. Okay? So the very first thing that it needs to be able to do is to absorb that energy, and that's where we're looking at the two structures in the middle. Okay, that's these guys here. Okay, and what ends up happening is when it's in its lock washer position, the protein is folded, and actually I got that wrong. When it's in its open position, the protein is forced that molecule open, and it absorbs light. Okay, that light energy it can then transfer further on down the line to other molecules. Okay, how can we tell it's transferring that light? Well, if we get rid of where it's transferring the energy to, what we can see is something known as fluorescence. Okay? So in the process of isolating your protein, what you're going to do is remove those other pieces, and what's going to end up happening is in your final solution, you'll be able to observe a fluorescence, signifying that the protein is still folded and still functional. Okay? It's releasing that energy, and since it can't release it to another molecule, it releases it as fluorescent light, but it's got to get rid of it somehow. Okay? So your solution should have a light pink fluorescent tinge to it, which looks kind of cool. Taken a step further, that molecule in its open form is also a deep royal blue. Okay? It absorbs a lot of energy, and that's why it ends up showing up as a blue color. Okay? So your solution with the proper protein folding is blue and fluorescent. The whole goal of this lab is to go through and see how we can mess with the protein folding. What things do you think you could do to change the protein structure to change how it's folded? Good. We've got side chains that are acidic and basic. We could add an acid. That acid is going to protonate certain side chains, which means we take a structure that was perhaps ionic and have now made it neutral. That changes the intermolecular forces. If the intermolecular forces change, that changes how the protein folds. Okay? We could also add a base, which could do the same thing. It could take something that was neutral or charged and convert it the other direction. If our structure changes, then if our individual amino acid structure changes, that will end up having an impact on our overall protein structure and can cause the protein to unfold. If the protein unfolds, it no longer forces that molecule into the open position, and it clamps into the lock washer position, and in that case, it no longer absorbs light, so it's no longer blue, and because it never absorbed that energy, it no longer fluoresces. Okay? If you're really, really good or careful with this lab, what you can do is actually do these changes reversibly. You can add a little bit of acid to it to get rid of the blue color. How would we neutralize acid? We could add base, and as soon as you add the base, you can sometimes get that blue color and that fluorescence to come back. So it is a reversible reaction, but it really requires some care.